Thank you for the opportunity to speak here, and thank you for the introduction, Firoz. So today my topic will be to accelerate drug product optimization using the integrated program delivery, what we call translational pharmaceutics. So as you can see, we are all trying to make drug development happen, get the nice molecules from discovery into market. And you always wonder how hard is it, but number of people that are working on drug development, you can say that it must not be that simple a problem. Then the question comes up, are we really configured to succeed in this business of drug development? And so we look at some of the challenges that we face today. At Quotient, we think about how we can operate differently. Is there a way to do things which can get us to market faster? Or is there a way to do things differently by understanding our challenges and addressing them? And that's where we come at translational pharmaceutics, where we try to apply different principles to re-engineer the drug products. And the drug product optimization might be needed for controlled release, modified release, or for solubility enhancement. And we all have probably seen both of these case studies. And thanks to Joe, we already saw so many challenges that we encounter with some of our molecules. And so today, focus will be on modified release development. So when we look at our industry, we can call ourselves fortunate because we have so many molecules that are entering clinical development. And there are so many companies trying to make a drug product out of it. However, the molecules are quite complex, and the physiology of the GI tract is not that easy to deal with the number of changes that happen. And so with the increasing complexity of development, we also face some failures, and the failures can then lead to increased development cost. And so the question again comes up, how do we try to address this? So there are multiple ways we can try to think about it. So one is to understand our molecules early, and that's where we try to characterize our molecules for solubility, permeability, DCS classification, BCS classification, and all those measurements can help us understand where our challenges could be. The other way to do it is to simplify our outsourcing and supply chains, and then build integrated models and processes. And so all of these lead to some kind of innovation in how we do things. And so let me talk about some of the industry structures that we have seen for several years. And so if you see on this slide, the two vertical rectangles are talking about functional expertise areas, the left one being the drug product development and the right one being the clinical testing. So we talk about CROs and CDMOs, and each of those boxes within their own functional area of expertise, they work very well, and the integration is extremely good. But when it comes to going from drug product manufacturing to clinical testing, that's where a lot of white space or gaps seem to be happening. And so we lose time and precious API sometimes during this transition. And so at Quotient, this is where we are trying to make an impact and try to be creative and innovative. We are trying to merge those two boxes and try to make it an integrated development platform for developing drugs. So on the left-hand side, we have formulation development, or a CDMO capability, where we have ability to manufacture on small scale or large scale various different product types, whether they are solubility enhanced, standard IR products, or complex modified release products. We have the capability to vertically integrate within that space, as well as use our CRO capabilities in both UK and in the US to integrate the testing of those products which are developed in actual human healthy volunteers. And so this actually is kind of a disruption of the conventional industry structure, where we combine both those activities into one company. And what that means is we are integrating formulation development, on-demand product manufacture, and clinical testing. A study was conducted at the Tufts University Center for Drug Development, where a comparison was made of studies which were conducted by using conventional process versus the translational pharmaceutics approach. And what they concluded was there was an average time saving of 12 months or greater. There was a consistent operational team that goes across the entire program, so that helps in keeping supply chain and product supply under control. Then we have significant reduction in R&D spending because we make very small batches and we generate very small fit-for-purpose stability data. So that saves API and therefore cost of the API. It is better decision making because we are using human data to generate the PK profiles, and that actually helps in using human as a model for the product that we are developing. And so there is greater likelihood of success. So with that, I just wanted to explain how is actually this acceleration achieved. So if we think about the process, on the right-hand side, there is a schematic which shows we do product manufacture, put it in healthy volunteers for testing, do the bioanalytical sample, get that data, and do the PK parameter estimation to generate plasma concentration time profiles. And so this cycle is what we call rapid manufacturing and clinical testing, and it used to be called rapid fact in earlier years when it was first introduced. So the main parts of the acceleration are achieved through drug product manufacturing integrated with clinical testing. The product is made in real time. That means if I'm going to dose the subjects next week or the following week, I'm manufacturing the product this week. And this allows us to make very small batch sizes. We also make stability data very fit for purpose, as I said on the earlier slide. And so this is reducing the API quantity significantly. So my batch size could be 250 to 300 tablets or capsules, and I will just make it in advance with limited stability of maybe 7 to 30 days. So in a conventional process, what happens is if you are trying to develop the drug product, a formulation analytical development group will start working on it. They will create several prototypes. Those prototypes will be tested in vitro assays as well as in some preclinical models. And then a few prototypes from those will be selected. Then they will be put on stability for a couple of months. And once the stability looks good, then some of those prototypes will be then scheduled for PK study in the human. The regulatory application is made after obtaining the data on the stability batches. On the right-hand side, you will see a graph of some of the preclinical correlations that were made back in early 2000. And that shows basically a scatter plot where you cannot know whether a dog is a good model for your molecule or a rat is representing your bioavailability predictions. So given that, and given this paradigm of testing, what happens is you're only able to test pre-selected prototypes in the humans. And if that prototype does not work, then you have to go back into this process again. So basically the process is delayed if your first 2 or 3 prototypes that you select for human testing do not make it. And so the last bullet says clinical testing is restricted to predefined products. And so when we see this as a challenge, at Quotient, we talk about drug product optimization by using translational pharmaceutics. What that means is we are going to do the same thing. We are going to do formulation analytical development at the early stage. We are also going to generate CMC data for regulatory submission. However, we are not going to make big batches. We are not going to be limited to 2 or 3 prototypes. We are going to develop something called design space. And by that, we mean we'll try to have a flexibility in the types of formulations we make. So let me see which part. So all the product optimization of the release profile. So we will develop a design space. So what I'll get to the next slide to explain that concept. But we do that design space development. We generate CMC batches, and then we make the regulatory application. And then you have already seen this schematic on the earlier slide where we'll make those prototypes in real time, dose them in the subjects, get bioanalytical data. The PK team is already ready by this time to generate the parameters. And then looking at this data, the client team and the Quotient team will decide which is the next product to manufacture. Do I want to optimize the release rate, or do I want to change the dose in the product? So this cycle will go on for 4 or 5 different periods, and then we'll be able to get our desired release profile. So as I said earlier, drug product optimization can be either for solubility enhancement or for modified release development. And so at Quotient, at Quotient, we have worked on multiple programs where majority of the programs have been for solubility improvement, and the next highest number is for modified release development. And then there are some other applications that we also work on. Within modified release, you can see there are different technologies, and Joe again referred to those technologies, and the choice can be based on multiple factors. And in solubility enhancement, we can also use different platforms, and generally we try to let the drug properties decide what platform we would use. So let's go back to modified release. In modified release development, typically, as we talked about earlier, we would develop 2 or 3 prototypes. One could be a shorter release profile, and the other one could be a longer release profile. So we develop prototypes for 4 hours, 8 hours, or 16 hours. And then we keep the dose the same so as to keep the surface volume ratios proper and keep the tablet size the same. This is how a typical formulation development would start. What we have noticed is whenever this kind of product comes to clinical testing, the typical challenge is that C12 improvement is not as good as the Cmax reduction. And you can see that in this graph. When we had 3 different types of profiles, the Cmax reduction is quite drastic, but the C12 improvement is not that great. And so we might end up losing a lot of AUC, if you want to think about it that way, overall exposure in the data that we get. And so when we do in vitro testing, we have no idea how this is going to perform in vivo. So this lack of confirmed IVIVC actually drives us sometimes into a second cycle. And if we have to go back into this cycle, you know that we would go back to basics and start formulation development again. And this is avoided by the design space. And earlier slide, I didn't explain what the design space was, so I'll try to give you a better picture of that. So when I said we develop a design space to get flexibility, what it is that we want to have flexibility in formulation. So let's say I'm developing those 3 profiles, 4-hour to 16-hour profile. I'll have a variable one, which is the release rate, and the other one would be dose. So I'll try to develop a design space where I can manufacture anything within this space and make stability batches at these 4 corner points. And I'll confirm that by making those 4 corner point batches and a couple of batches in the center here. What that does is I have stability data for 7 to maybe 30 days on these corner points, and we make a submission to the regulatory authority to dose any of these compositions in the clinic. We then don't have to make any amendments to the protocol. We write a flexible protocol that allows us to dose any of these compositions. And so we are able to navigate the space in this way. So let's say that we design a 5-period clinical study where we initially dose the IR product and get an idea of what the issues are. So you can see here we are trying to stay within the therapeutic window, but I'm getting above the safe dose level. So I want to reduce my Cmax. Typically for modified release, we are trying to eliminate the Cmax effects. And then my next iteration in the formulation will be, I want to give one of the prototypes. Say I'm giving an 8-hour release profile prototype. It gives me this profile. It has reduced the peak, but I'm also not using the full therapeutic area window. And so the next prototype, we get together as a team and then we design, okay, I think we could increase the dose in this case and then try to see if I can get my profile to match my expectations. And so this is how we would navigate within the design space, and the clinical study design would guide us to do that. So this is a real case study where the compound was being dosed twice a day. They wanted to eliminate the side effects due to Cmax. And we did exactly what I described in the previous 2 slides. We generated data for 14-day on the corner points and in the middle of this design space. A matrix type approach was used. The Cmax of the IR product is shown in the red, and then the optimization was carried out using the clinical design where we thought we might need 5 periods. So one would be given for a reference IR product, and the remaining periods could be used for the modified compositions of the matrix tablet. But what we noticed was at prototype 3, period 4, we were able to get the profile that was desired. And so 5th period, which was already built into the study, was then used for a food effect understanding of the same dosage form. Let me just point one more thing. So when we talk about acceleration, what you can see here is that the product was developed in 3 periods, and the lack of food effect was established, and all this work was conducted from start of formulation work to clinical report in 7 months. This is another case study where this particular situation, the compound again had a BID dosing. They wanted to remove the Cmax related problems and then try to match the C trough levels to the 60 milligram BID dose. And so in this case, they didn't want to stick with one platform. They wanted to evaluate matrix mini tablets as well as monolithic matrix tablets. And so that space was designed for both those platforms. And then the clinical study was designed where matrix mini tablets over a certain window of release rate was compared to the unit dosage forms. And then comparison was made for food effects, doses, and so on. And then finally, they picked the dosage form that they wanted to choose based on the two. They liked the matrix monolithic tablet better than the mini tablets. In the third case study, we have multiparticulates. And here, again, same issue, Cmax reduction and reduced dosing frequency. So in this case, we developed the multiparticulates using extrusion spheronization, and a functional coating was applied. So in this case, we had three variables to look at. One was the ratio of the immediate release to the sustained release pellets. The second was the release rate, as we have talked about on the matrix tablets. And the third one was the drug dose. So again, we have a 5-period study design, and starting with understanding the first prototype, then dosing the reference, and the other prototypes, changing the ratio of IR to SR, and trying to adjust the dose. We were able to get the steady state PK predictions that were done by both. First, we did modeling and simulation to get the predictions, and then we showed that through clinical testing, we were actually able to match the numbers for the prototype 3, and so that product was going forward. And in this particular case, the timeline for formulation start to decision-making data was about 9 months. This is a case study which is quite a complex case study, but what I want to show from this example is essentially every compound is unique, and the requirements for getting a certain target product profile are very different. And so in this particular case, we know that drug overdose and sometimes intentional, sometimes accidental is a major issue with pain medications. And so this is a technology which the company Enseiss is working with. And they have a multiple pill abuse resistance strategy along with a trypsin activation abuse protection. So they have a complex system here because there have been several programs that have tried to prevent drug overdose but have met with limited success. So this is one of their approaches to try to overcome this problem. And so they have a prodrug which requires activation by trypsin in order to release the oxycodone. And then inhibition of trypsin in the GI tract actually can prevent the absorption of oxycodone. However, the inhibitor that is used for inactivating trypsin is nafamostat, and that has a very short half-life. So then they had to develop an ER formulation to make sure that the nafamostat is released over a period of time that matches with the amount of time the prodrug is going to stay in the GI tract. So just stay with me. I know it's a little complex, but it gets better. So the one part of the study was to optimize the nafamostat beads. And similar to the previous situation, we are trying to make the beads using, using the concept of IR plus SR. But in this case, the beads are made by drug layering on cellets. And so there are immediate release beads, and there are SR beads. Within the SR beads, there is a question about, do I work with weight gain or do I work with the polymer ratios to optimize the release rate? And so in this case, they found that weight gain had to be kept constant and the polymer ratios had to be varied using different polymers. Again, the design space was developed for that purpose. And then the question to be asked was, what is the dose of nafamostat that is going to be adequate to let 1 or 2 tablets work well? But if more than 3 tablets are taken by the person, then it has to act to prevent the conversion of the prodrug into oxycodone. So there was some work done to understand the clinical design. And on this slide, it's basically just showing the design space, making sure that we are able to reproducibly make the batches for the different coating levels or different compositions of coating material. And the second parameter was the dose of nafamostat, which was in the range of 0.25 to 35 milligrams. So several PK studies were conducted to understand the relationship between the prodrug conversion to oxycodone, and the presence of IR plus ER beads of nafamostat that were going into the study. So after identifying the ratios that were necessary, it was shown that in the second part of the study then, it was shown that when 1 to 3 tablets were taken, the levels of oxycodone were fine. But if you take any more than 3 tablets, then the mechanism of trypsin inhibition would kick in, and then the nafamostat ER plus IR beads would start working, and they would prevent this excess oxycodone levels in the blood. So doing this complex design, they were actually able to get through to the point where they can now understand what nafamostat dose is required, how much PF614 gets converted to oxycodone. And this prodrug cannot be taken by any other route because it needs activation in the GI tract. So essentially, it's an oral prodrug that delivers oxycodone in right doses and prevents misuse, either intentionally or an accidental case, there won't be any toxicity. So they are trying to address this, and they got, the company got breakthrough status for this particular development of the technology. So in this case, the conventional formulation development and manufacture would have taken several years to develop this complex concept. And in this particular case, the entire program from formulation development to demonstration of these protection properties was completed in about 19 months. Throughout the program, the first initial dosing started within 6 months, and then 11 data-driven formulation assessments were completed in about 10 months. So you can see the speed at which program could be conducted once you have a clear idea of what question are you trying to ask. What is the question about the drug product, and what are you asking in the clinic to answer for you. So this PF614, the prodrug, and the abuse resistance multiple pill could be the first opioid product and which has the abuse resistance and overdose protection. So in summary, I hope I was able to get through to you that while drug development is increasingly complex, and it will probably remain complex, I think we just need to think about ways to disrupt this situation and find areas where we could make an impact and try to get successful in our attempts to take individual drugs and try to understand their properties to finally make it into commercial success as well as address patient needs. We think that translational pharmaceutics accelerates formulation development. It provides formulation flexibility, and the integrated clinical protocols enable data-driven decision-making. And this decision-making is using human data, sometimes because preclinical data does not necessarily guide us in the right direction. I went one point ahead, so basically it's on clinical data, and the impact of poorly predictive in vitro and preclinical models can be mitigated. The design space strategies can be effective in modified release, and I just showed you several case studies where the in vitro in vivo correlation could be inadequate, and so we try to get the correlation by actually dosing it in the clinic. So that's where I would stop. I'll try to see if I can answer some of your questions.
Any questions for Aruna?
Yeah. So thanks. That was a great presentation. I had a couple of questions. So you did mention when you're developing the design space and the fact that you're actually using like humans as part of the study. Could you comment on the actual number of runs in your DOE or any kind of model that you use to narrow down the number of people that are actually involved in the initial one, and also comment on the cost of actually
Yeah.
doing this.
So the first part of the question was number of
Number of runs. So I'm
runs.
thinking if you're having 2 or 3 factors.
Yeah. Yeah, so number of runs, as I showed, a 2D design space allows us to make, like, we generally make 4 different formulations and then 1 or 2 formulations in the center. The number of runs, typically, clinical study cannot be more than 5 or 6 periods because you have to get the same subjects to come back. A typical study would have between 12 and 16 volunteers. Our medical team is very careful with, the safety of the substance that we are trying to dose. So they will first look at all the properties of the molecule, what safety data they have in the clinic prior to dosing a modified release product in the clinic. So 12 to 16 volunteers, they can come back, so you don't get data in the same subjects, so you're eliminating the variability coming from different subjects every time. So generally between 3rd and 4th period, we are able to get a good formulation, and then we can either drop the last period if it is a 6-period study, and if it's a 5-period study, we generally get a food effect out of it. And then some clients like to do a multiple dosing to see where we can reach steady state with the control release. So initially we can start with one clinical study and 1 formulation optimization, and then add pieces on if the data seems to be promising. And a lot of times we go to the second part. Did I answer your question?
Yeah. And then the second one was the cost. So
Yeah, cost. Toby, do you want to take that cost question?
Sure. Yeah, hi, Toby with Quotient Sciences. For the type of study depends on the number of formulations vertex we're going to develop, and then also the clinical scope. So typical studies about $1.5 to $2 million US.
Including the farm dev and the clinical testing. Right? Yeah.
All services in data science.
Yeah, data sciences. Actually, that is another thing. We have extremely good data sciences group. They can do data collection and data analysis for PK and taste assessments, whatever. They can do everything, so start to finish. So we have a lot of small company clients who like to take advantage of those services because they then don't have to think. So it's truly integrated program where once you have the drug and what questions we want to ask in the clinic, we can really design from start to finish.